Good morning, students. Today, let us discuss about cleavage. After fertilization, which takes place inside the ampular region of the fallopian tube, a diploid zygote is formed. The zygote then passes through the fallopian tube to reach the uterus. As the zygote passes through the fallopian tube, cleavage takes place, that is, repeated mitotic division. Cleavage is the repeated mitotic division of zygote to form a solid ball of cells called morula, which later changes into a hollow ball of cells called blastonia or blastocyst. The difference between cleavage division and normal mitosis is there is no period of growth in between the divisions. During cleavage, the size of blastomeres decrease as their number increase. The cleavage is holoblastic, that is, it divides the zygote and blastomeres completely into daughter cells. This is the zygote, which is surrounded by the zona pellucida. The first cleavage is meridional, that is, from animal pole to vegetal pole, and produces two daughter cells called blastomeres. We can see that one cell is slightly larger than the other. The two blastomeres remain adhered to each other. The second cleavage occurs at right angle to the plane of the first. That is, it is an equatorial division, thereby forming four cells. The large cell divides a little faster than the other, hence a transitory 3-cell stage is seen before the 4-cell stage of the embryo is reached. The third cleavage is meridional, thereby forming 8 cells. The division continues until 18 or 32 cell stage is reached. The solid ball thus formed is called morula. The morula consists of 16 to 32 cells, but the size is same to that of zygote. The morula, if you dissect, can see is made up of two types of cells: outer layer of small, clearer cells and inner layer of large cells. The morula reaches the uterus about 4 to 6 days after fertilization. It still remains surrounded by the zona pellucida, which prevent the sticking of the morula to the uterine wall. The morula, when reaches the uterus, its outer layer of cells absorb a nutritive fluid which is secreted by the uterine mucous membrane. Due to the influx of the fluid, the outer cell layers become flat and is called trophoblast or trophoectoderm. The inner layer of cells arrange themselves at the animal pole, forming a cavity called blastocele or blastocyst cavity filled with the fluid. As a result, the inner cell mass gets separated from the trophoblast except at the animal pole. The inner cell mass looks like a small knob. This knob gives rise to the embryo and hence is known as embryonal knob. As the quantity of fluid increases, the morula enlarges rapidly and assumes the shape of a cyst. Now it is known as blastocyst. The trophoblast cells which are in contact with the embryonal knob are known as cells of Ruber. The blastocyst, when reaches the the morula which is converted into blastocyst, now attaches with the uterine wall. The zona pellucida, which surrounds the blastocyst, guides the attachment of the blastocyst to a proper site of the endometrial lining where implantation can take place. The portion of the blastocyst where the embryonal knob is located lies against the endometrium. The contact stimulates the proliferation of the trophoblast cells. These cells secrete and release lytic enzymes which destroy the endometrial lining, and the blastocyst enters into the pit that is formed due to the corrosive action of the enzyme. The blastocyst sinks and then gets completely buried in the endometrium. The corroded part is repaired. After implantation, the blastocyst forms a connection with the maternal tissue by forming finger-like projections which in turn digitate with the maternal tissue to form an organ called placenta. Hence, in this way, a zygote from single-celled undergoes cleavage to form a multi-celled, and in this way the blastocyst gets implanted into the uterus for the further development. Thank you.